Welcome. Today we are going to talk about vitamin D binding protein concentrations quantified by mass spectrometry. Definition: vitamin D is important for good overall health and strong and healthy bones. It's also an important factor in making sure your muscles, heart, lungs, and brain work well and that your body can fight infections.
Especially when it comes to your health. That's why we want to break it down bit by bit and talk about the little things that slip by sometimes. Today, it's vitamin D. Let's get into it. Vitamin D belongs to the fat-soluble family of vitamins. Fat-soluble vitamins are vitamins that get stored in your liver and fat tissues when your body has more than enough of them already. Fat-soluble vitamins are important to good health, but you'll find that you don't need a ton of them to stay healthy, especially if your body is letting them stick around for a while. So let's talk specifics about vitamin D. Vitamin D plays a big role in your body's use of calcium and phosphorus. It helps by increasing the amount of calcium that can be absorbed by the small intestine, and that means good news for forming and maintaining healthy bones. Vitamin D is especially important for children to develop strong bones and teeth. So here's how you can get it. There's a reason people say milk makes your bones strong, and let me tell you, it's partially to do with the vitamin D. Other dairy products might also be vitamin D fortified, so keep an eye out for those. Oily fish will provide vitamin D, so that's salmon, sardines, and cod liver oil, which is not for everyone, I must say. You can also get vitamin D from ultraviolet light, as in the sun, so 10 to 15 minutes of sunlight is a great way to get outside and get some vitamin D. So how much do you need? The recommended dietary allowance for vitamin D is measured in micrograms. How much you need depends on your age. From 1 to 50 years old, the recommended daily allowance is 15 micrograms. After exceeding 50 years in age, 20 micrograms of vitamin D is recommended. If you're not getting enough vitamin D, there can be some repercussions associated with bone development and maintenance, such as rickets and osteoporosis. So that's vitamin D. If you have anything you want to add, don't forget to join the conversation in the comments. That's all for now. Thanks for watching. If you enjoyed this video,
Application in medicine. More than 50% of Black Americans have a diagnosis of vitamin D deficiency, yet their low concentrations of 25-hydroxyvitamin D do not appear to convey a higher risk of fractures as they do in white Americans.
Vitamin D has a significant effect on 229 genes, supporting its disease-preventing potential. Vitamin D has a significant effect on at least 229 genes, some of which have been associated with Crohn's disease and type 1 diabetes, according to UK and Canadian researchers. The research, published in Genome Research, found 2776 binding sites where vitamin D attached to the genome, many of which were concentrated around genes that have been linked to autoimmune conditions such as multiple sclerosis and rheumatoid arthritis and diseases such as colorectal cancer and lymphocytic leukemia. The researchers said the findings backed the role of vitamin D in maintaining health in a world where 1 billion people are estimated to be vitamin D deficient due to either diet or lack of sunlight. There is now evidence supporting a role for vitamin D in susceptibility to a host of diseases, said lead author Dr. Sriram Ramagopalan from the Wellcome Trust Center for Human Genetics. Vitamin D supplements during pregnancy in the early years could have a beneficial effect on a child's health in later life. Some countries such as France have instituted this as a routine public health measure. The researchers isolated fragments of DNA bound to vitamin D receptors, VDRs, treated them with vitamin D form calcitrol, and then sequenced the DNA fragments. VDRs are proteins activated by vitamin D that attach to DNA and influence which proteins are made from our genetic code. In this way they identified the 2776 vitamin D binding sites, which Dr. Ramagopalan said illustrated the importance of vitamin D in gene expression. The research was funded by the Medical Research Council, MRC, the MS Society, the Wellcome Trust and the MS Society of Canada and conducted by the University of Oxford. Vitamin D status is potentially one of the most powerful selective pressures on the genome in relatively recent times. Our study appears to support this interpretation, and it may be we have not had enough time to make all the adaptations we have needed to cope with our northern circumstances, said one of the authors, Professor George Ebers, from the Action Medical Research Professor of Clinical Neurology, in reference to skin shade and sunlight exposure.
Pros and cons. Pros. Mass spectrometry gives information about the molecular weight of your compound and when performed in conjunction with a combustion analysis. The
mass to charge, they're deflected the least, whereas the brown, they're deflected the most. They're the ions with the lowest mass to charge ratio. As they reach the detection plate, they create a small electric current which is amplified, and this produces the stick diagram that you see in the spectrum on the top right. The more ions of a particular mass to charge ratio, the greater the current, and the taller the stick in the spectrum. Therefore, the taller the stick, the more abundant that ion of that particular mass to charge ratio. So there's the red going through. It would represent, I suppose, the Gulf ball in our earlier analogy with the moderate mass. So what are the summary points or key points? Tiny amounts of sample, very tiny, are required for mass spectrometry. The sample must be changed to a vapor. So if it's a solid or liquid, it has to be heated. One or more electrons are removed from the atoms or molecules to produce positive ions. An ion accelerating electric field, together with plates that have slits, form a narrow beam of positive ions. The magnetic field deflects the ion beam, or the ion. More ions.
Cons. Some other disadvantages include that you will not be able to distinguish between isomers of a compound having the same charge to mass ratio. Separating enantiomers usually requires something such as a chiral column. Issue applied in Ecuador. In Ecuador, there are no cases related with experiments of vitamin D binding protein concentrations quantified by mass spectrometry.
2.2. The mass spectrometer. The mass spectrometer is a vital instrument in many kinds of chemical analysis. But how does it work? And how do we interpret the data that we get from it? A mass spectrometer works in 5 stages. First, the sample is vaporized. Then, it is ionized. Third, it is accelerated. Fourth, it is deflected. And fifth, it is detected. The deflection stage is the heart of the mass spectrometer. When we accelerate an ion, it would like to keep moving in a straight line. This tendency of the particle to move in a straight line, it's
Conclusion. Since GC1F is more common among blacks than among whites, it is unclear whether vitamin D binding protein levels differ according to the race.